Hello class. Now that we've talked about the structure of the membrane, now let's talk about transport processes that occur in the cell and across the cell. So our first process is diffusion. We're going to think about what's going on with diffusion, what influences the rate of a diffusion flux, and why diffusion is well suited for short distances only. So diffusion can occur through a tissue, it can occur across the membrane, then we start bringing in the membrane into it, and what happens with the diffusion of water? A process called osmosis. How do you know what direction that it's going to occur in? And what are the factors that influence osmosis? One of the things we need to do is distinguish between osmolality, which is the number of particles total, and tonicity, which is the number of impermeant particles or non-permeable particles, non-penetrating particles. So we want to think about various scenarios, thinking about taking red blood cells and placing them in various solutions. Then we're going to move on to facilitated diffusion. Facilitated diffusion is still a passive process because you're using a gradient, but you need a transporter to help the substance get across the cell. So it's not really diffusion because facilitated diffusion can saturate. It can reach a transport maximum, and so that makes it not really diffusion, even though it's called facilitated diffusion. And then we're going to bring in active transport, and active transport is using ATP as an energy source instead of just the energy in the gradient. And there are some proteins that use ATP directly for primary active transport, and some proteins that use the gradient that was generated by primary active transport. So here we go. This is a picture of two solutions. One is a glucose solution, one is pure water, and there's been some kind of pretend barrier in between them, and suddenly the barrier is removed, and now glucose is able to diffuse along its concentration gradient. So you have to realize that molecules are moving all the time, they're colliding all the time, and when you suddenly remove the barrier here so that glucose can travel, glucose has an opportunity for fewer collisions over here than over here. And so glucose will gradually bounce around and go from the area of high concentration to an area of low concentration. And that movement will continue. You'll get to the point where both compartments have the same concentration, but movement still continues. But at this point we have no more net diffusion. So if we look at the concentration gradients of the two compartments, one that had 0 glucose and one that had 20 millimolar glucose, you see that diffusion is not a linear process. It happens very quickly in the beginning and gets slower and slower and slower, and then at some point they reach a point where the diffusion gradient is gone and both compartments have the same concentration. And notice both compartments end up with half the concentration, and that works because we have two compartments of the same volume. In our subsequent examples that may not be the case. So we have this beautiful equation that dictates diffusion flux, and this isn't an equation that would describe this this process. This would be the diffusion flux over time, and that's not exactly what I'm talking about in this next equation, and that's for all you engineer types out there that are used to a different equation. Those of you that don't know what I'm talking about, ignore me. Okay, so the flux in moles per second is related to a number of factors. So first of all, what is the substance? What's its permeability? What's its molecular weight? Those kinds of things are in this constant. What's the area of diffusion? The larger the area you have for diffusion, the faster diffusion will occur. What's the concentration difference? The bigger the concentration difference, the faster the flux. And what's the distance? The farther the distance, the lower the flux. So you take these units and you plug them in, and you see that we get moles per second equals moles per second. I'm not going to ask you to calculate anything with this. I just want you to think about what's the substance, what's the area, what's the concentration difference, what's the distance. So the more permeable, the more soluble something is, the faster it'll diffuse. The bigger the area, the faster it'll diffuse. The bigger the concentration gradient, the faster it'll diffuse. The longer you have to go, the slower the diffusion. Other things that influence the flux, what's the temperature? Faster temperature means more kinetic energy and more collisions. What's the nature of the gradient? And how big is the gradient? How big are the particles? Bigger particles are going to diffuse slower. What's the surface area? More surface area, more diffusion. Think about the lungs. The lungs have a huge surface area. It's an adaptation that promotes a greater diffusional flux for oxygen. What's the distance? The farther the distance, the worse diffusion is. If you want to take a molecule of oxygen and have it diffuse from the alveoli to a capillary in the lungs, beautiful, very short diffusion distance. If you want oxygen to diffuse from your lungs all the way down to your big toe, that's going to be thousands and thousands of hours. So thank goodness we don't have to diffuse oxygen down to our big toe. We have a blood system that carries it down there. What's the medium you're diffusing in? Gas diffusion is faster than liquid diffusion. Liquid diffusion is faster than a colloid, or say something like cartilage or a jelly-like substance. So what's the viscosity? And then when you introduce a membrane, what's the characteristics of your membrane? Something cannot diffuse across a membrane if it can't get through the lipid membrane, or it doesn't have a channel to get through. Alrighty. So water can diffuse, and that's called osmosis. So here we have a volume, and it's all water. So we've divided it up into little squares, and each one is a little water molecule. And then we have some solute over here. So notice that as the solute concentration goes up, the water concentration goes down. So technically, we talk about osmosis as the diffusion of water along its concentration gradient. So, if we had a semi-permeable membrane in between that allowed water to move, this area has a higher concentration of water than this. So maybe you've seen experiments before with a U-shaped tube, and there's a little dialysis membrane or a selectively permeable membrane. We have pure water on one side and some kind of sugar solution on the other side. Does it tell us what this thing is? We don't know what it is. Let's say it's sugar. Well, the water concentration is higher on the left arm, so water will osmose to the other compartment and the level of the fluid will rise. Now, if we took a little piston and pushed on here, how much pressure would we have to exert to prevent the fluid from rising? In millimeters of mercury would be our osmotic pressure. So the osmotic pressure would be the difference in your water concentrations. Whoops. Here we go. Now, if we talk about a situation portrayed here, we have 3 little red blood cells, and they're in a big beaker. This is a little bit different situation than our original diffusion because we had 2 compartments that were the same volume. This volume is teeny, 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 tiny as compared to the bath. So the red blood cell will not change the concentration in the beaker, but the beaker could change the concentration in the cell. Now that's why we have to be clear about what situation we're dealing with. So, if I have an isotonic solution, not just the number of particles, but fixed particles, if I have an isotonic solution, my little red blood cell is happy. Water's coming in and out, but there's no net movement. If I have a hypotonic solution, it means water is entering the cell more than it leaves, and entering so much that it undergoes hemolysis. Now, remember red blood cells are these little discs? They can actually blow up to a sphere. So their volume can double. But water gets more, you have more water than double the volume, then the thing is going to go kerplooey. And we call that hemolysis. A hypertonic solution would be more concentrated in terms of particles, less concentrated in terms of water, and water would leave, causing the cell to shrink, and that process is called crenation. Now, these three scenarios involve particles that are fixed. The particles cannot move. Only the water can move. And water is able to move because red blood cells have an abundance of a particular kind of protein called an aquaporin, which is essentially a water channel. So water can easily move between the inside and the outside, and whatever the particles are in our solutions cannot move. The particles are impermeant or fixed or non-penetrating. But what if the particles can also move? Well, then we come to something called tonicity. Tonicity is not just the total number of particles, but the total number of non-penetrating particles. So we have 400 non-penetrating particles. That's hypertonic. That will cause the cell to shrink. We have 200 non-penetrating particles. That's hypotonic. The concentration of water is higher, so water enters and the cell swells. So osmolarity is the measure of the total number of particles in a solution. Tonicity measures the number of impermeant or fixed particles. And it's the fixed particles that you have to compare to consider where something's going to shrink or swell. So here's some examples of problems. Here's what you have to think about. Are the substances going to dissociate? Are the substances penetrating or non-penetrating? Now, you can work through some of these problems on your own, but I have an activity that we will do in class, and we'll work on this together. So I'm not going to tell you the answers today, but this is how you work through it. Sodium chloride. Sodium chloride is going to dissociate. So 180 millimolar sodium chloride will be 160 milliosmolar sodium chloride. Inside the cell will be 300 milliosmolar. So you have 160 on the outside, 300 on the inside. Where's the highest concentration of water? Well, it's going to be outside the cell. What's water going to do? Water's going to osmose into the cell. What's the cell going to do? It's going to swell. And 160 is about half of 300. And I would suspect that those cells would swell so much that they would even burst. If this were only 100 millimolar, the cells would swell and maybe possibly would not burst. So, work through that example on your own. Try these other examples, and we're going to work on applying this in class. Now, some things cross the membrane along a concentration gradient, but they need a transporter. We call that facilitated diffusion or facilitated transport. Now, the people that named this probably shouldn't have named it this, because it's not true diffusion. The reason why it's not true diffusion is because the process saturates. It saturates at a Vmax. Vmax is a transport maximum. What's the molecular basis of saturation? Well, the molecular basis is how many transporters do you have? So here's an example of a facilitated transporter. We have a high concentration outside the cell, a low concentration inside the cell. that solute binds to the transporter changes shape and allows the substance to be carried in. The rate of transport should increase as your concentration gradient increases, but you'll get to a point where the outside is so concentrated and the speed of transport can't increase anymore. You've reached a transport maximum. And why is that? It's because you're using all of your transporters. Here's another nice example. Here's glucose binding to a GLUT transporter. The GLUT transporter changes shape, and glucose is brought in the cell. Now here's a cool thing. Glucose is immediately phosphorylated, and now it's no longer glucose, and we maintain our inward concentration. However, this transporter saturates at about 10 millimolar. So glucose is going to go up and go up, but once you get to 10 millimolar glucose, you're using all your glucose transporters, and you have saturated this process, and so no more glucose can come in. And so since this transporter saturates so quickly, we usually think of glucose as a non-penetrating solute because it's so slow to come in. And if you're having trouble with that, ask me in class, and we'll go over that. So here you see that transport phenomenon. With a typical diffusion situation, and you have your solute concentration the more the concentration, the more the flux. Now notice this is linear, but we're not comparing time, we're comparing solute. The diffusion of a substance is not linear with time, but that's not what we're plotting on this graph. And again, if that's not tracking for you, please ask me. But what you notice here is that when we bring in a mediated transport process, it levels off. And when you're leveling off, it means all of your transporters are occupied. All of your transporters are working as fast as they can. And even though the concentration gradient has increased, you can't go any faster. So the reason why facilitated diffusion is not true diffusion is because it saturates. Now anything that involves a transporter can saturate. So we also have transporters that are involved in primary active transport. Here you see this transport pump. It's taking 3 sodiums and taking it out of the cell, taking 2 potassiums and taking it in the cell, and we need to phosphorylate this thing to get that to happen, and so it requires energy. So the sodium-potassium ATPase is located in just about every cell of the body. It's running 24 hours a day. It's a big part of your basal metabolism. And the purpose of this pump is to establish a differential distribution of sodium and potassium across the cell, such that you have high sodium outside a cell and high potassium inside the cell. Here's another version of that pump showing you 3 sodiums going out and 2 potassium going in. So this is an electrogenic version of this pump. There are some sodium-potassium ATPases that are 1-to-1. Now, sodium-potassium ATPase establishes a sodium gradient. Then you can use the sodium gradient to transport other things. So for example, sodium comes in and calcium goes out. So this is a sodium-calcium antiporter or a sodium-calcium exchanger. A lot of these are found in heart muscle. It's a way to keep calcium low. Here we see sodium coming in while hydrogen goes out. There are a lot of these transporters in the proximal tubule of the kidney. Notice that the 2 molecules are going in opposite directions, so they're called antiporters or counter-transporters. Those are examples of secondary active transport. ATP is not needed directly, but ATP was needed to make the sodium gradient, and then the sodium gradient drives this transport. Sometimes the substances can go in the same direction. So here we have a sodium glucose transporter called an SGLT transporter. Sodium and glucose are coming in together, so symport or co-transport. And then here we have a sodium and an amino acid. So sometimes you can have facilitated diffusion and secondary active transport in the same cell, and I'll show you an example of that in the next in the next presentation. So we have talked about diffusion. Oxygen and CO2 can diffuse across a plasma lipid bilayer. When we have aquaporins, we can talk about the diffusion of water across the membrane in osmosis. And when we have transporters, then we can have the transport of ions across the membrane. Charged ions are never going to go through the bilayer, but they can use proteins to get across the membrane. This transport can take place along a gradient with facilitated diffusion or against a gradient, either directly with the help of ATP or indirectly with a sodium gradient made by ATP. So think about the presentation, think about your learning objectives, figure out what's making sense, make a note of what's not making sense, and we can talk about it in class. There's only one presentation more to go in cell physiology. Thanks for hanging in there with me, and I'll talk to you next time.